So this is a short video on a cute gram-negative bacilli, that is Pseudomonas aeruginosa. Let us try to understand it through this pictorial representation. The first thing is about the basic morphological characteristics of Pseudomonas aeruginosa. So there are four basic characteristics of Pseudomonas aeruginosa. First one being the fact that it's a gram-negative bacilli, which is suggested by the fact that the bacilli drawn here has got a pink shade there, suggesting that it's a gram-negative, and the shape of course suggests it's a bacilli. The second point is that it's a non-encapsulated organism. So there's no covering to this bacilli, which suggests that it's a non-encapsulated organism. The third point is it being a strict aerobe. So that's suggested by the fact that there's an oxygen mask that is connected to the face mask, which is connected to an oxygen cylinder. The fourth point, the basic point regarding its morphology, is that it's motile organism with the flagella there. So those are four basic morphological features of Pseudomonas aeruginosa. Once we have understood those four basic features, let us understand about the four pigments that Pseudomonas aeruginosa produces. They are pyocyanin, pyoverdin, pyrubicin and pyomelanin. Right? So that is suggested by the speech bubble here, wherein the Pseudomonas says, I play holy every day with red, green and brown. So this again is to emphasize the fact that it produces these pigments. Okay, so it produces this pigment, so it plays holy every day with these pigments. So those were four basic things, the morphological characteristics of Pseudomonas aeruginosa and four pigments that Pseudomonas aeruginosa produces. Now let us try to understand the culture medium which is required to grow Pseudomonas and the biochemical reactions that are characteristic of Pseudomonas aeruginosa. Right? So to make it easy to remember them, we have depicted them in a form of diet chart. So diet chart of our Pseudomonas. So there are three biochemical diet that Pseudomonas loves to have. So what are those three biochemical diets? So the first one being the oxidase ka bartha, which suggests the oxidase test being positive for Pseudomonas. The second one is the hug-Leifson soup, which is basically hug-Leifson media in which Pseudomonas oxidizes glucose. The third thing is the catacola, which is nothing but the catalase reaction, again which is positive for Pseudomonas aeruginosa. So that was about the biochemical test for which Pseudomonas shows positive reaction. Next, you have the culture diet. So what are the culture diets? Culture diets of Pseudomonas aeruginosa. There are 5 in number. The first one being the nutrient shake, which is basically suggesting the nutrient broth in which Pseudomonas will form a surface pellicle, because we knew that it's a strict aerobe, right? So hence it forms this pellicle, surface pellicle in this nutrient shake. Next, you have the nutrient agar cake, which is suggestive of nutrient agar medium, in which Pseudomonas has characteristic features. So like it produces the sweet odor and green-blue color of the pyocyanin pigment. That was the second culture diet. The third culture diet of Pseudomonas is the red currant pancake, which is suggestive of the blood agar, in which Pseudomonas exhibits beta hemolysis. Right? So that was the third diet. Next diet is the McConkey sauce, a yellow McConkey sauce, suggesting that the McConkey agar will remain yellow despite the growth of the Pseudomonal colonies there. That's because it does not belong to Enterobacteriaceae family, right? So it's a yellow McConkey sauce. And the most favorite dish, the culture diet of Pseudomonas aeruginosa, is the citrimide Karela fry. Now the citrimide Karela fry basically is nothing but the citrimide agar. And why do we label it as the most favorite diet of Pseudomonas aeruginosa? That's because it is the selective media for Pseudomonas. That means it selectively allows the growth of Pseudomonas aeruginosa, the citrimide agar or the citrimide Karela fry. So this was a short video on basic points regarding Pseudomonas aeruginosa that are to be written when you're asked to write about Pseudomonas aeruginosa.